Welcome to the second video for Chapter 2. We'll start by finishing Section 2.1. Our microscopes have the ability to magnify and resolve images. Magnification means that the lens is able to enlarge an image, right? It makes it look bigger relative to the actual size. We often see on our eyepieces or ocular lens that there's something written on the side, such as 10x or 10 times. That means the lens is able to make the object appear 10 times larger than it actually is. Resolution is the ability to resolve the image, the ability to distinguish two very closely spaced objects as separate. And I have two images shown here. Greater resolution is shown on the right? That's higher resolution, where you have a sharp image, the better ability to distinguish two very closely spaced objects, versus on the left, we have a low resolution image, right? It would be blurry. We probably wouldn't be able to tell that they're two separate objects if they're very close together. There are two factors that impact resolution. The first is wavelength, and shorter wavelengths or higher energy light provides better resolution, clearer images. And that's why electron microscopes, we're going to learn about in a bit, are more powerful than our typical light microscopes, which use longer wavelengths or lower energy light. The second factor is numerical aperture, abbreviated NA, and there's no units for this. It's dimensionless. It's a measure of the lens's ability to gather light. So imagine these are different objective lenses, and the numerical aperture is usually written on the side of that objective lens. And I draw a normal line in the middle. The numerical aperture is a measure of the amount of light it can gather, but it also describes the range of angles over which the lens can gather light. So you can see it can gather light at this angle, but it could also gather length at a shorter angle. Whereas this one has a low numerical aperture on the right because the angles of light over which the system can accept the light is very narrow. Even when a microscope has high resolution, it can be really hard to tell apart the different structures within the specimen, especially if they're transparent, if they're see-through. So we need to increase the contrast, which is the difference between the specimen in the background or the difference between different parts within the specimen. Something else we're going to practice during the first several weeks of lab is staining. We're going to learn different types of staining techniques that use dyes to either distinguish the specimen from the background, or dyes that bind to specific structures within the cell in order to identify those parts. In summary, remember that magnification makes the image or the object appear larger. Resolution allows us to see fine details so we can distinguish structures within the specimen. And contrast enhances specific features, either distinguishing the specimen from the background or specific structures within the specimen. And we need all three to produce clear, informative images through our microscopes. All right, another checkpoint. Magnification versus resolution. So magnification was the ability to enlarge the appearance of something, right? Make it look bigger than it actually is. Resolution, in contrast, is the ability to distinguish two very close objects as separate entities, as separate things, right? It determines the image clarity and detail. The second one is similar, so we're looking at resolution again. Resolution is how clearly small or very closely spaced structures can be seen as distinct, whereas contrast is looking at the visible differences between the parts of a specimen, right? Letting certain things stand out, like the specimen compared to the background, or certain structures within the specimen compared to another structure, especially when the structures or the cells are transparent. Two factors that affect resolution. We talked about wavelength. Remember that shorter wavelengths provide better resolution, and numerical aperture, which is usually written on that objective lens. The higher the numerical aperture, the better the resolution. That takes us to the end of section 2.1. Gather your notes and try to answer these learning objectives in detail. Briefly, let's look at these. Identify and define the characteristics of EMR. Remember that EMR, or electromagnetic radiation, is a type of energy that travels in waves. including things like radio waves, microwaves, visible light, UV, and more. So in microscopy, we can use visible light and UV light to visualize really small things. Remember that EMR is characterized by things like wavelength, frequency, and energy, and look at the relationships between those. Remember that shorter wavelengths are going to provide you with greater resolution, such as UV light, compared to visible light. For the second one, how lenses are used in microscopy to manipulate visible and UV light, remember that light, when it passes through lenses, like the convex lenses in our microscope, will be bent due to changes or differences in refractive index. We use convex lenses to converge light to a focal point so we can magnify the specimen. And remember that the curvature of the lens and how the distance from the lens to the object will change how the image appears and its magnification. Some microscopes take advantage of UV light, specifically those fluorescence microscopes. Remember, we can use UV light to excite fluorescent dyes, and that will ultimately lead to the emission of visible light as their excited electrons fall back to the ground state. In section 2.2, we'll examine the history and individual contributions to the invention of simple and compound microscopes. Fracastoro was an Italian scholar similar to those that I mentioned in Chapter 1, in that he proposed disease could also spread through invisible particles he called seminaria. He proposed that these particles could stick to things like our clothes and be transferred between individuals to make them sick. But again, just like prior cases in Chapter 1, his theory also remained hypothetical because there was still not enough or insufficient technology to observe these microorganisms. There's no evidence that they existed yet. Leeuwenhoek is credited with being the first individual to build a powerful, simple microscope and to observe these microorganisms. He was a fabric merchant or a Dutch cloth maker, and it's said that because they had to use magnifying lenses to view the quality of the fabric, he ultimately became able to develop these really advanced lens making techniques. What's different between Leeuwenhoek and others who were building simple microscopes at the time is that he shared his observations with the Royal Society of London, which ultimately gave him the credit for the microscope and observing these organisms. There were other early microscope contributors as well, including other eyeglass makers in the Netherlands, as well as Galileo Galilei, who is famous for his telescopes, but also used a compound microscope with two lenses to look at insect parts. Here's Leeuwenhoek on the left, who's credited with creating the first simple microscope, shown here, where he saw those animalcules and wee little beasties. Imagine if we kept this name animalcules, then microbiology would be called animalculology instead of microbiology. And even though these were really simple microscopes, remember simple means we have one lens compared to compound microscopes like that created by Galileo, which has two or more lenses. Even though these were simple, they were actually very powerful, right? With even better resolution than some of the compound microscopes at the time. Robert Hooke also made important contributions to microscopy. He used compound microscopes to look at different specimens, and he published his findings in a book, Micrographia. He was also the first person to use the word cell based on his observations. So he described the structure of cork he saw under his microscope and saw these structures that looked like small boxes or honeycomb-like structures that turned out to be, now we know today, dead cells with just the cell walls remaining. So his work and his publication really helped establish the foundation of cell theory. And cell theory includes, from his observations, that cells are the basic units of structure and function in living organisms. Here's an example of his compound microscope and a page from that book where you can see the cork that he saw with those honeycomb-like or cell structures. This is not in our textbook, but I wanted to share with you that the Royal Society of London, that National Academy of Sciences and pretty much the world's oldest scientific academy, actually published Hooke's book, Micrographia, online. So here's an article about the publication. Here is a screenshot of what you'll see if you go to his book, and you can click through the pages and see all the neat images that he included, what he saw. Here's our next checkpoint. What's the difference between simple and compound microscopes? Remember that simple microscopes use a single lens to magnify something, such as Van Leeuwenhoek's microscopes. Compound microscopes use two or more lenses in sequence to achieve a greater magnification, and Galileo and Robert Hooke were examples of individuals who used compound microscopes. If I compare and contrast the contributions of these different individuals, Van Leeuwenhoek, remember he developed that first simple microscope. He had advanced lens making techniques. He was the first to see those single-celled organisms, microorganisms. He sent his observations to the Royal Society of London, which really gave him that scientific recognition. Robert Hooke, he used compound microscopes to look at natural specimens. He published Micrographia in 1665. He coined the term cells by looking at those cork and other plant cells. Galileo, remember he used his technique from telescopes to create the first compound microscope, and he used that to study insect anatomy. The end of this section examines who actually invented the microscope. While Van Leeuwenhoek and Robert Hooke usually get the credit for their contributions to simple and compound microscopes, nobody can really know who actually invented the first microscope. Some argue that it's the Jansens. These were Dutch eyeglass makers who might have invented these microscopes. The problem is that they were really secretive about their work, and they didn't really have any records of their work. It could also be their neighbor that invented the microscope or the telescope. Van Leeuwenhoek really gained recognition after his friend basically convinced him to submit his work to that Royal Society of London. And it's through that evidence, through that documentation, that really made him famous. Same thing with Robert Hooke. By publishing his work in Micrographia, he really became well known and also gave others the interest in microscopy. All right, let's recap section 2.2. Looking at the historical developments and individual contributions, so maybe the Jansens or Lippershey should be credited with the first microscope, but nobody really knows because there is a lack of documentation there, right? A lack of evidence. So Van Leeuwenhoek is really credited with that first simple microscope with great lenses. He was the first to go see those single-celled organisms because he's documented and shared his findings with that Royal Society of London. Robert Hooke also documented his work and published Micrographia, what he saw through his compound microscopes. He also coined that term cell. Looking at simple versus compound microscopes, remember simple microscopes use a single lens to magnify something, like Van Leeuwenhoek's microscope. Compound microscopes use two or more lenses in sequence to achieve a greater or higher magnification and resolution. So these were used by Robert Hooke and Galileo. Simple microscopes are easier to make, but more limited in what they can magnify or how much they can magnify, whereas compound microscopes give you greater image clarity and probably better detail when you're visualizing these small structures. All right, section 2.3 really gets into the details of microscopy. We'll look at the different parts of microscopes as well as all of the different types of microscopes we have. We'll start with light microscopes, which use visible or UV light to make images, and we're going to discuss these different types of microscopes. Light microscopy does not include electron microscopes, which use short wavelength electron beams instead of light, and they also do not include scanning probe microscopes, which function how they sound. They use probes that are passed over the surface of the specimen. The bright field microscope is probably the most commonly used type of microscope. It's what we have in our labs, and it's probably what you used in your previous courses as well. So this is the most common type of compound light microscope, and it produces a dark image on a bright background. They can be either monocular if they have only one eyepiece, but these days we usually try to stick to binocular light microscopes with two eyepieces. Each eyepiece has an ocular lens within it that usually has a 10 times magnification, although this can vary and it's written on the eyepiece. And we also have objective lenses. Usually there are several of these that are mounted on a rotating nosepiece. And they generally vary from 4 all the way to 100 times magnification. So on our microscopes, we'll see 4, 10, 40, and 100 times objective lenses. One important calculation is the calculation for total magnification of a microscope. To do this, you just take the ocular lens magnification and multiply it by the objective lens magnification. For instance, if you were on the objective lens, the 40 times objective lens, which is blue usually on our microscopes, and you multiply that by the 10 times ocular magnification, you get a total mag of 400. I'm going to use an image of the microscope to describe this next part. When you're viewing a specimen under the microscope, you'll need a glass slide, and you typically place your specimen in the center. Then you take your glass slide and you're going to want to set it on the stage by opening up the stage clip and clipping in your slide right there, and it's held in place right here. There are XY knobs underneath the stage, either on the left or right, depending on the type of microscope you have, that will allow you to move your specimen either left and right or forward and backward, which is basically toward or away from you. When you're using the microscope and you're starting to focus your specimen, you're going to use coarse or fine focusing knobs. We usually start with the coarse focus or the coarse adjustment knob for these lower objective lenses, either the 4 times or the 10 times objective lens. These are going to be the shorter objective lenses that are found on your revolving nosepiece. These will make large movements to the stage, so the stage will go up or down in pretty big movements. Whereas when you're using the bigger, the stronger objective lenses, either the 40 times or the 100 times objective lenses, you need to start using the fine focus knob, which is the smaller one, outside of the coarse knob. These make very small adjustments to the stage. So you're moving the stage much more slowly. And this is important because, let me make some space here. Let me erase this. Using the fine focus knob is important when you get to these higher magnification objective lenses, is because once you're using the greater magnification, like the 100 times, it's going to be super close to your glass slide. If you accidentally were to use your coarse knob, then you could potentially push the objective lens into the slide and crack it, which would shatter glass and maybe put live bacteria into your eyes. I'm going to use the same image to describe this part as well. As you view your specimen under higher and higher magnification, you'll notice that brightness is reduced, so you'll need to increase the intensity of your lighting. In our bright field microscopes, there is a light source or illuminator towards the bottom of the microscope beneath the stage. Light will then pass through a condenser, which does what it sounds like. It condenses this broad beam of light into a narrow, strong beam of light, focusing it through the specimen. This condenser is controlled by a condenser focus knob, which is usually somewhere on the side, and that can adjust the height of the condenser, moving it up or down for optimal lighting. You can also control the brightness by either adjusting the rheostat. The rheostat adjusts the intensity of this light, making it brighter or dimmer. If you ever turn on your light switch and you don't see any light, maybe change the dimmer switch, this rheostat, to see if someone turned that all the way down. You can also adjust light intensity, let me erase some of this, by changing the opening, the iris diaphragm. So the diaphragm is shown here, and there's a little knob here where you can make the iris bigger or smaller. So you can have a bigger beam of light shining through your specimen, or you can close the iris diaphragm to have a smaller beam of light pass through. very similar to how the iris of your eye changes the size of the pupil. We can view specimens under the bright field microscope because light is transmitted, absorbed, reflected, or refracted by structures within the specimen. Later on, we're going to use stains, and stains contain chromophores or pigments that affect how light interacts with the different structures within the cell. We use them to improve contrast and resolution of our image. So remember, in bright field microscopy, we're going to see basically dark or colored specimens against a bright background. And the limit of our magnification using these microscopes is about 1,000 times. That means we can see up to small bacteria, but not viruses. Viruses are too small to be seen with our light microscopes. As we get to greater and greater magnifications, imagine you have your 100 times objective lens and your slide here. Air gaps between the lens and the slide can cause blurriness. It can change resolution, really worsen it. And that's because of that refractive index difference between the air and the glass slide. So what we often do, or what we usually do, is use our oil immersion lenses, which are, in our case, the 100 times objective lens. And we're going to use immersion oil here to fill the gap. So there is going to be no air space as you view the specimen. Oil is similar to glass in its refractive index, so light won't bend as much, and more light will be able to collect or be collected by the objective lens. You'll have greater resolution. There are different types of oils we can use depending on the light, and we'll see these in lab. Here's just air on the right if you don't have oil when you're working with the oil immersion objective lens. What happens is that the light scatters, right, because of the different refractive indices. And overall, imagine you have a broad beam of light, well, most of which is not being captured by that objective lens. You'll get a blurry image, low resolution. In contrast, when you have the immersion oil, there's less or no scattering. Usually there's a tiny bit, but most of the light will be captured by the objective lens, and you'll get greater resolution and a clear image. You have to remember that when you're adding oil, there should be no space between your slide or the cover slip and the objective lens. The objective lens has to be submerged into the oil for this to work. Please take some time to review the best practices for microscope maintenance. Make sure you're always cleaning the lenses with lens paper after use. Let's see, don't use the coarse knob for those higher magnification objective lenses. Make sure you clean the oil from the oil immersion lens and check the other lenses. Here we have an additional resource if you'd like more practice learning the parts of the microscope or viewing virtual slides under the microscope. And that takes us to the end of our second video. We'll finish this section in the next video.